Good morning, everyone. We are the Group 2 of BIO20-1. We are about to discuss our topic, which is the introduction to biosensors and bioreceptors. The biosensor. A biosensor is an analytical device that allows it to interact with the analyte. It consists of two components, the bioreceptor and the transducer. A bioreceptor is a biomolecule that recognizes the target. The transducer, on the other hand, converts the recognition event into a measurable signal. It is considered to be an enzyme of sorts, and this is what the biosensor is after. These bioreceptors detect analyte. Now, what is this analyte I keep on mentioning it before? An analyte is a chemical substance that has been identified and measured. However, analytes themselves cannot be measured as a whole. Thus, their physical and chemical properties can only be measured by each. For example, a table is an object, however, on its own cannot be measurable. However, you can find the table's height, width, and length through proper measurements. That is the analyte. Now for the transducer. A transducer, or also known as the detector element, transforms the signal resulting from the interaction of the analyte with a biological element into another signal that can be easily be measured. In layman's terms, it is a device that converts variations into electrical signal. In this topic, there are 5 types of biosensors. Firstly, the calorimetric biosensor. It measures the temperature change of the solution containing the analyte and interprets it in terms of its concentration. Secondly, the potentiometric biosensor. This biosensor uses electrodes to convert the biological reaction into an electrical signal. It mostly uses a pH meter glass electrode for its own detection. The acoustic wave biosensors are coated with antibodies which bind the antigen into the solution. This reduces their vibrational frequency and is used to determine the amount of antigen. The AMPEROMETRIC biosensor, on the other hand, uses current signals that are applied by 2 electrodes. These biosensors are used to measure redox reactions. Lastly and finally, the optical biosensors measure both catalytic and affinity reactions. They measure the change of fluorescence and change induced in the intrinsic optical properties of the biosensor itself. This sensor was used on fireflies for the catalytic reaction.
Hi. Now I'm about to discuss history of biosensor and bioreceptor. But how biosensor and bioreceptor started? Well, it won't be possible without Professor Leland C. Clark Jr., who first introduced the biosensor concept. He is also known as father of biosensor. In 1956, he published his definitive paper about the development of an oxygen probe. He addressed his desire to go beyond the limited range of analytes that could be measured in our body. In 1962, he expanded the idea of more intelligent electrochemical sensors at the symposium in the New York Academy of Sciences. His concept was illustrated through an experiment where glucose oxidase was entrapped at the Clark oxygen electrode using dialysis membrane. Clark and Lyons coined the term enzyme electrode, which was expanded to build functional enzyme electrode. In 1969, Gilbolt and Montalvo introduced the potentiometric urea electrode. They use potentiometric measurements on liquid electrode with urease to measure urea concentration. In 1974, this was the year where thermal transducers for biosensors were proposed, such as thermal enzyme probe and enzyme thermistors. This is an example of thermal transducer. In 1975, Clark's idea became reality. Why? Because Yellow Springs Instrument Company launched the glucose analyzer, which is based on the amperometric detection of hydrogen peroxide. This was the first of many biosensor-based laboratory analyzers to be built by companies around the world. In the same year, Davy suggested the use of bacteria as biological element in microbial electrode for the measurement of alcohol. Lubers and Pupids coined the term optode and described a fiber optic sensor, which is an indicator used to measure carbon dioxide or oxygen. In 1976, Clemens et al. Incorporated an electrochemical glucose biosensor in a bedside artificial pancreas, which is marketed with the name of BioStator, that today is now not commercially available anymore. Laroque introduced the lactate analyzer LA640, in which soluble mediator and hexacyanoferrate was used to shatter electrons from lactate dehydrogenase to an electrode. In 1982, Shichiri et al. Reported in vivo application of glucose biosensors, which is the first needle-type enzyme electrode for subcutaneous implantation. This is an example of glucose biosensor. In 1984, first mediated amperometric biosensor was introduced. Ferricin used with glucose oxidase for glucose detection. In 1987, Medicine's Exatec launched blood glucose biosensor and a pen-sized version of it. This is an example of the product. In 1992, iStat launched handheld blood analyzer, which make it convenient now for people to analyze their blood. This is an example of the product. We just put the blood sample at the bottom of the iStat handheld blood analyzer. In 1996, home blood glucose monitoring found its success because it reached $175 million in sale. This is the example. The biosensor. In 1998, launch of LifeScan fast-take blood glucose biosensor. This is the example of the LifeScan blood glucose biosensor.
Hi, good day. My name is BIO20-1, and today I'll be talking to you about the applications of biosensors. But first, a little recap. Biosensors are an analytical device used to detect analyte. It is composed of a transducer and a bioreceptor. A bioreceptor is a material with biological elements that lets it interact with an analyte, while a transducer converts signals coming from the interaction between the analyte and the bioreceptor. They are widely used and very helpful in the field, research, and industry. Biosensors are now considered an important part in the research field industry of namely food, medical, and environment. They provide better stability and sensitivity as compared with the traditional method. Biosensor is backed up with technology, and that's why they can provide better stability and sensitivity, and they, as time passes by, they can improve. They are cost efficient. Biosensors, one of its best advantages is they are cost efficient, because biosensors are less expensive to make and less expensive to operate. Some biosensors in the field of food. Using biosensors to detect pathogens in food. Pathogens is one of the cause of illnesses and food poisoning in food. It uses E. coli, a bioindicator of food contamination. That is, E. coli is found in vegetables and can be extracted. Once extracted and it has reacted with the analyte, the potentiometric alternating biosensing system will detect the variation of pH caused by the ammonia, the antibody of E. coli. So it can detect pathogens. Second is determination of artificial sweeteners. Artificial sweeteners. Artificial sweeteners are considered bad because they are addictive and they coax people into eating high energy food, which will cause obesity and malnutrition. This biosensor is combined with lipid films with electrochemical techniques. It detects the electrophysiological activities of the taste epithelium. An instrument called spatiotemporal technique is used to detect the reaction between a taste cell and the sugar, which will then determine what kind of sugar it would be. Some biosensors on the field of medicine. Glucose monitoring. Glucose monitoring is one of the widely commercialized biosensor in the market. It is used in hospitals with diabetes patients and other glucose-related illnesses. This biosensor uses the enzyme glucose oxidase to break blood glucose down, which will produce FADH2. Then FADH2 will be oxidized by the electrode. The electrode, that is the transducer, will then produce a current which a device will read so that it can determine the blood sugar level of a person. A fluorescent biosensor is an imaging agent used for cancer and for drug discovery. This biosensor uses small scaffolds where fluorescent probes are mounted, and when these bioreceptors approaches the analyte, the scaffolds will produce a fluorescent signal that can be detected and will be recorded. Sensors in the field of environment. Microbial biosensor. Microbial biosensor can detect damaging bacteria present in air, food, or water. This biosensor uses oxygen microelectrode with microbial cells immobilized in polyvinyl alcohol, has been fabricated for the measurement of bioavailable organic carbon in toxic sediments. This biosensor has also been developed to detect BOD, a total amount of organic material present in wastewater. Immunoassay biosensor. Immunoassay biosensor can detect low concentration level of pollutants. This biosensor uses immunoelectrodes as its transducer and uses pesticide-specific antibodies to detect the presence of low concentration pollutants. Other applications of biosensors. Food safety and quality. Determination of toxicity and toxicity levels. Nanobiosensors. Biodefense biosensing. Industrial processing and monitoring. Biosensors in plant biology. To summarize my report, application of biosensors is an important part in gathering data to be used on various fields, on various purposes, and it gave way to a different and efficient way on gathering data.
New biosensor and bioreceptor technologies. Sweat biosensor. Sweating from exertion or a stifling room temperature can be impractical for some patients, and unsafe for others. And unless they are on second leg of the Tour de France, it's unlikely patients will want to sweat all day for the benefit of a sensor reading. But researchers at the University of Cincinnati have came up with a novel way to stimulate sweat glands on a small, isolated patch of skin so subjects can stay cool and comfortable and go about their daily routine without spending hours on a treadmill. UC professors came up with a device of a band-aid that uses a chemical stimulant to produce sweat, even when the patient is relaxed and cool. The sensors also can predict how much patients sweat, an important factor in understanding the hormones or chemicals the biosensors measure.
Kidney biosensor. Indian Institute of Technology at Bombay and Indore have jointly developed a biosensor that makes it possible to detect kidney disorders in less than 8 minutes. The biosensor can accurately measure both the pH and urea concentration with a single drop of urine. Researchers who have developed the biosensor believe that it will help make a point-of-care test to ascertain if kidneys are functioning normally. For a kidney function test, doctors need to estimate of pH and urea, as most kidney disorders result in reduced pH and higher concentration of urea.
Cancer biosensor. In order to determine if at what levels certain biomarkers are present in your cancer, your doctor will need to take a sample of tumor tissue or bodily fluid and send it to a laboratory to conduct a series of advanced pathology and molecular profiling tests. Those tests will detect and measure the levels of your cancer's specific biomarkers.
Drug detector. Stanford engineers create experimental technology to monitor and maintain drug levels in the body. The new technology has three basic components: a real-time biosensor to continuously monitor drug levels in the bloodstream, a control system to calculate the right dose, and a programmable pump that delivers just enough medicine to maintain a desired dose. The sensor contains molecules called aptamers that are specifically designed to bind a drug of interest. When the drug is present in the bloodstream, the aptamer changes shape, which an electric sensor detects. The more drug, the more aptamer change shape.